Hey all, it's Dr. B. This is a short video to go over section 2.4 of genetics and genomics in medicine, the different um, databases you can use for a lot of genetic information. I'm going to spend um, all this time on just two of them. There are a lot. These are ones that I would argue are used more than just about anything else. So I'm going to start from the beginning here, and then I'm going to have you do a little web quest, and in this Edpuzzle video, you'll be able to answer some questions. So first thing you basically do is just type NCBI, and you'll see the National Center for Biotechnology Information. You click on that, and then click here. This is basically a repository for a lot of information, both protein, nucleic acid, genome related, homology related, literature, you name it. This is used a lot. And basically, I'm just going to show you a fundamental use for it. And then later on in the semester, you'll be doing an assignment using this in part. So let's just take any random gene that you're maybe aware of. Like, for example, P53. So if I just type P53 and I hit search, you can see there's a lot of information on it. And of interest, there are 365,097 documented papers on this, or connections to papers on this. There have been 35,242 genes that have been submitted by people. Now you may be saying, well, that's just one gene. Shouldn't the sequence be the sequence? This is the number of people that have inputted their sequence that they found in here. And again, many of these will be completely redundant. And this is the idea, is that when you find a DNA sequence, you can submit it to NCBI, and it will just add to the accuracy of it over time. So there's a lot more here. You can see that as far as nucleotide, let's just click on nucleotide. And you'll see here that this is a lot of the different submissions that people have had for the different sequences of P53. And it's not, by the way, you'll notice that I didn't specify this to be human. So mus musculus, that's a mouse. So that's their particular P53, a piece of mRNA, CDS, the coding sequence of it, and all kinds of stuff. And you can go ahead and just click, Homo sapien if you want to see the human. Here is human, and somebody just submitted one for exon 8, so let's just randomly just grab that one and see what that's like. And so this is so a couple things about how to read this. This is the identifying, it's called a session number or GenBank number. This is its identifier, and it's 137 base pairs long, and it's DNA and it's linear, and it was submitted on July 25th, 2016, and it was submitted by this group right here, and it was part of a paper they did on this. As you can kind of see here how that works. So as you go down here, there's different versions of it. This is the little bit of this particular exon that they found the sequence to be. And so there, of every 10 letters, there's the DNA that they found for it. And so from that, there's a couple other things of interest, one of which is called FASTA, or FASTA format. This is just, when you click on this, it's basically the straight up DNA that we can submit for things like comparison and whatnot. So that's one use that it has. It also has another use in the idea that you can, if you have a DNA sequence and you don't know what it is, you actually can use NCBI to figure out what it is. Like, I'm going to show you one right now. So like, what if you had a piece of DNA and you want to know what it was? Well, you can use something here called BLAST. And there's different kinds of BLAST. There's a nucleotide BLAST, a protein BLAST, other kinds of BLAST. We're just going to go to nucleotide BLAST. And basically what I would do here is I'm going to paste some DNA. Now, maybe that's from something that I sequence, and I don't exactly know what it is. Maybe it's a bacteria, and I sequenced, 16S gene. I have no idea what it is. If I pop it in there, don't worry about all this stuff yet, and I just blast it, it's a verb that basically means it's comparing that sequence against every other sequence it has in the entire database to tell me how similar it is to other stuff. And so you can see here that this DNA appears to be from something called Amblyomma quadricavum, which, by the way, I happen to know is a tick that's located in Puerto Rico. And you can see here that it's 100% identical to it. And so the way that I can use this is really nice. I can also do a graphical summary of it, and I can also do an alignment. So I can use this to compare the sequence, excuse me, sorry, I said a little coughing fit. So basically you can see here that the query is what I put in. This is this top row here, that's what I put in. And the bottom one is the subject. That is what is in the actual database itself. And you can see that this one is 307 of 307. It's exactly aligned, so that's how they know that that's exactly what it is. However, if I move down, let me move down to something that's not the same. Let me just turn off all these and compare our query to, for example, this thing. And we can do an alignment of that. We can see it's not 100% aligned. Only 86% of these are aligned. And you can see that right in this position in my query for the quadricube is an A, but in the same position in one of the samples in the database was a G. And so that is, generally speaking, kind of how it does what it does. It does a lot more than this, but I wanted to kind of give you guys a little bit of introduction to something that if you're going to be in the field of molecular biology, you're going to use this. So I'm going to ask you two little questions here, and we'll see how you do.